Hi, I'm Ariel Howell. I'm will be the moderator today for our recorded webinar session with Malaria Partners International and partnering with the Seattle Malaria Clinical Trial Center. Joined today by Dr. Kublin, the medical director of this program, and Dr. Murphy, the clinical investigator. I was first introduced to Malaria Partners International through my work with Interact and Rotaract throughout my high school and undergrad time, and now I'm a community member of a Rotaract club here in Seattle, and I've been a board member on Malaria Partners International for about a year and a half. I've been very grateful for my experience with the board to hear more about how malaria impacts the world globally and specific populations that are most impacted by this disease. Malaria is considered one of the greatest killers of humankind, and in 2019 alone, 229 million people had cases of malaria, and of those, 409,000 people died from a preventable and treatable disease. Most of these people were young children and pregnant women, and 94% of these cases occurred in sub-Saharan Africa, which is where a majority of our work actually takes place and is focused. Malaria Partners International is a nonprofit organization founded and run by Rotarians, and we're working to ignite an international campaign amongst Rotarians for the global eradication of malaria. A lot of our work is focused on giving presentations to advocate for the end of malaria at local or international Rotary Club meetings and Rotary conferences throughout the US and Africa. In addition to this, we provide funding through our small grants program to Rotary clubs around the world that want to get involved in the fight to end malaria as well. Again, I'd like to introduce Drs. Kublin and Murphy of the Malaria Clinical Trial Center here in Seattle to hear more about how they work to recruit healthy volunteers to solve and work towards a variety of ways to treat and investigate malaria, and hear more about ways that our board or local members can get involved and just learn about their research a little bit more. So I'd like to invite you to take it away.
So welcome everyone. My name is James Kublin. You can call me Jim. I'm the medical director of the Seattle MCTC. I'm based here at the Fred Hutch Cancer Research Center, where we've been conducting what we call controlled human malaria infection studies, or CHMI, for well over a decade.
So I'm Sean Murphy. I'm a physician scientist as well. I work at the University of Washington in the Department of Lab Medicine and Pathology, where I'm a clinical pathologist. My research lab, which is located in South Lake Union, works on malaria vaccines and diagnostics. And I moved here in 2008 in large part to work on malaria vaccine trials with Jim. And we've been working together ever since. I've been working on malaria for more than 20 years.
All right, well, I'll start a slide deck that provides a bit of a snapshot and overview of the Seattle Malaria Clinical Trial Center. And today we'll be just briefly introducing you to the Seattle MCTC and the type of work and studies that we do there. Overall, our mission is focused on testing the safety and efficacy of malaria therapeutics and vaccines in humans in IRB approved, that's essentially the ethical review and approval of, and FDA reviewed and approved clinical trials. We have principal partners as part of the Seattle MCTC that includes Fred Hutch, the University of Washington, and Seattle Children's Research Institute. And I'll describe a little bit more of each of their roles in the Seattle MCTC. But first, a brief introduction to the CHMI, or Controlled Human Malaria Infection model that we use to test these drugs and vaccines. These are primarily focused on early phase studies of drugs or vaccines to select candidates for further advancement into very large late phase field studies. And these large field clinical trials, of course, typically are conducted in malaria endemic areas. So prior to the challenge or infection of an individual with malaria, we administer a drug or vaccine and then infect individuals with malaria either by mosquito bites or needle-based delivery. And this is a tremendous tool that can further advance malaria drugs and vaccines. Other pathogens, serious global pathogens such as HIV and tuberculosis, do not have the benefit of this CHMI model, and significant delays result from the need to go through very large clinical trials in the field before one even has a signal of efficacy of a drug or vaccine. So this has proven to be incredibly effective for malaria vaccine and drug development, and also has a tremendous and excellent safety record. And essentially, this cartoon highlights how we conduct these CHMI studies. We have a registry of volunteers actually here in Seattle, of very motivated, predominantly very altruistic individuals who are interested in contributing to the advancement and ultimate eradication of malaria. And typically we randomize these individuals to either the placebo or an active intervention, such as an immunization or a drug. We then infect the individuals with malaria and follow them after that infection to assess whether or not the drug or vaccine works in controlling the infection, or whether they develop malaria infection as revealed by the parasite in the peripheral blood. Dr. Murphy, or Sean, has made tremendous advancements in detecting that infection in the peripheral blood that has really advanced the CHMI model and made it even more safe and well controlled as compared to in the past. So this is another cartoon of the standard CHMI design. On the upper half of the slide, you see the cartoon of the preparation of infected mosquitoes, which takes about 8 weeks prior to actually infecting an individual via that mechanism of infected mosquito bites. The parasite develops in the mosquito, it migrates to the salivary gland, and then infects an individual. When that bite occurs, it takes about 7 days for it to multiply until it emerges into the blood or erythrocytic stage that you see on the right. And ultimately, a thick blood smear would be positive several days later. I alluded to Sean's more advanced technique detecting these parasites with molecular methods, and that detection of the parasite is typically 2 to 3 days earlier than one would see the parasite on a typical thick blood smear. And on the lower half is simply a cartoon of a typical clinical trial design in which we have screening and eligibility assessments. We do the CHMI challenge. We have then outpatient follow-up, sometimes a hotel phase and treatment, further outpatient follow-up, and typically the end of the study. This can take anywhere from 2 to 6 months for the total duration of one of these clinical trials. These figures and photographs illustrate the mosquito bite method of CHMI. Again, these mosquitoes are reared in the laboratory under very sterile conditions and are ultimately used to infect people with malaria. A more recent advance is using cryopreserved parasites that are isolated from the mosquito and directly injected into the vein via a method called DVI, direct venous inoculation. And this takes just a few minutes. It's very painless. It's less invasive than even a typical blood draw. So we've used this very effectively here in Seattle now for about a decade. And I mentioned Sean's approach to detecting the parasite in the blood using this molecular method. And this is something that we have applied to both drug and vaccine studies here in malaria. The first publication looked at a drug study that we were investigating, DSM-265, to assess whether or not this drug could prevent malaria. and found that the application of this assay or RT-PCR method really advanced the field with regard to making sure that we identified these infections very early on. And most people had no symptoms from the actual malaria infection because we were able to identify the infection very early on. Among those who did develop symptoms, they were generally mild, mild fevers, chills. And once we measure and diagnose the malaria, we administer the drug, and within a couple hours, they're symptom-free. So this has really improved both the safety and the efficiency of our clinical trials. The Seattle MCT organization is all based here in Seattle. I mentioned the Fred Hutch, where we have a prevention center, clinical trial site. We have a central institutional review board, which has reliance agreements with our partners. This is for the ethical and scientific review of our studies, and have a very strong regulatory and data management group. We have additional clinical sites at the University of Washington, as well as the vaccine trials evaluation units with the University of Washington and Kaiser Permanente. We have these mosquito bite challenge facilities, both at Seattle Children's and at the University of Washington, and laboratories that are supported by each of these partners to investigate safety, the malaria diagnostics that Sean leads at the University of Washington, sample processing, and intensive evaluation of the immune responses, both elicited by the vaccine, but also by in response to the infection itself, that may help inform us how to improve malaria vaccines in the future. This is just an illustration of the locations of our different partners. The Fred Hutch, as I've mentioned, provides study administration, clinical site, and processing support. The University of Washington South Lake Union campus provides the malaria diagnostic lab, as well as the CHMI site via infected mosquitoes. And Seattle Children's has also a long history of working with us, providing GMP or good manufacturing procedure insectary for these infected mosquitoes, and as well as other immunology and malaria assays. So an upcoming clinical trial we have that's starting just in a few weeks and will proceed through the autumn and early winter of 2021-2022 is focused on a 3-dose regimen of the PFSPZ vaccine. This is manufactured by Cineria. We'll be testing whether this 28-day regimen is tolerated and also is efficacious against a heterologous CHMI. And that means that the strain of the parasite used in the vaccine is different from the strain that we'll be using in the CHMI model, which reflects the more likely diversity of the parasite that one would see in a field setting. And we'll be conducting this CHMI or challenge at 2, 6, and 10 weeks after the last dose of the vaccine. This will really enable us to assess the efficacy of the vaccine early on after vaccination, at a midpoint or 6 weeks later, and at a more durable time point 2.5 months later. This provides you an example of the primary objectives of a typical study that we would do. This primary objective, of course, is focused on vaccine efficacy of this vaccine against Plasmodium falciparum malaria in adults residing in locations where malaria is not endemic, following heterologous CHMI. And then, of course, we always are very attuned and focused on assessing the safety and tolerability of any intervention that we're investigating at the Seattle MCTC, including vaccines in this case, and also drug studies. And this is just an illustration or figure that highlights the type of schedule that we would be following in a typical vaccine study. This is for the upcoming Cineria study we're doing here in Seattle this fall. As you see, it's broken down into these 3 different groups. They'll all be receiving the same regimen or immunizations with the same dose. And you see that they receive these immunizations on study day 1, 8, and 29. And then for group A, you see that the days to CHMI after that last vaccination is 14 days. In group B, it's 42 days. And in group C, it's 70 days. And this then is followed by a evaluation of the vaccine efficacy. We'll have a total number of 54 volunteers participating in this clinical trial. So how do we get people to volunteer to be infected with malaria? Most people have a bit of a nervous response to that. And again, we have a tremendous record of both safety and tolerability of the different drugs and vaccines that we're investigating, and a tremendous rigor of clinical monitoring and evaluation of our clinical trial participants. And so we have an extensive recruitment effort that highlights the burden of malaria worldwide, which continues to, of course, impact millions of people worldwide, and has almost 500,000 predominantly young people, children, perishing from this terrible parasite. So we have recruiting strategies that include flyers that we distribute at different venues. We have, of course, the internet or social media based efforts. We've used Facebook, as well as a number of other type of recruitment venues like Craigslist. We post all of our clinical trials on clinicaltrials.gov. And we also use radio and different other social media events to highlight the work we're doing, and in particular, when there's a clinical trial upcoming, to provide some description about what that trial is trying to accomplish. For example, looking at this upcoming scenario study and evaluating the efficacy of the vaccine at different time points after having received the regimen. And then a main tool that we use to bring participants to our clinical trials is a clinical trial registry of volunteers. And we have hundreds of people, again, locally in Seattle, who have expressed an interest to participate. But as you saw, the schedule of events for these clinical trials can be somewhat intense, and therefore many people may be interested to participate, but because of other commitments, cannot join a particular study at a particular time. So this is an area in which we really need more local support, more local enthusiasm to increase the number of interested volunteers that can join our clinical trials. And for more information, we've provided both mine and Sean's contacts information here. This is thanks to all of our many collaborators and funders that we've been working with over now more than a decade, and look forward to answering the questions that Malaria Partners International has for us, and also welcome Sean to contribute to any further discussion. Thank you.